my God, bro. This is so good.
It's so good.
You're watching masters at work here.
Yeah. There's something called a measurement problem in biology. I basically have to kill the cell, get all of its contents out, and then do RNA sequencing to figure out what kind of RNA is inside. So there's a lot of information loss, right? There's cellular decisions that happen, let's say now.
Yes.
And then the result is only going to come forward in a few days.
Yes.
Right? So how do I connect those two things?
Right.
And that's where the breakthrough comes in here. It's called time vaults, okay? Genetically encoded molecular archives. And it's repurposing something called the vault particle to physically encapsulate mRNA and store it as the cell just goes about its daily business.
We created almost a cellular backup system like Time Machine on your Mac.
Yeah. Dude, it's exactly that. There's a single protein that makes it up. That's called a major vault protein. You get 78 copies of this protein and it self-assembles. It's like a bunch of tiny little magnets. 78 of them come together and make this barrel shape. Beautiful structure, right? So how do we get from vaults to now time vaults?
Yes.
This is a really funny story, okay? So one of these students at the Harvard lab found Leonard Rome's YouTube channel. He's called the Vault Guy.
The Vault Guy.
On YouTube. And he makes a bunch of YouTube videos about vault proteins. Yeah, well, I guess one of these like grad students was like, yo, this Vault Guy is making some pretty cool stuff. Maybe we could use him for our purposes, right? And that's where they got the idea of time vaults. And then this paper has come out in Science, a genetically encoded device for transcriptome storage in mammalian cells. So how do we do that? How exactly mechanistically does it work? Yeah, like how am I going to get the mRNA inside? This is again at the molecular level.
Yes.
Okay. Well, at the molecular level, everything is Lego blocks. So one of the properties of mRNA is when it comes out, it has something called a poly-A tail, which means at the end of the mRNA, there's a bunch of AAA, the adenine. There's just repetition of adenine, kind of like a handle for figuring out where to take this mRNA and things like that. And what these guys figured is we could have a container and a bait. Okay. The container is my vault protein shell, and my bait would be this protein that interacts with the polytail and also has a little domain on the protein that interacts with the vault. Okay, so the PABP, that binding protein, is going to grab the poly-A tail, and then part of it is going to get stuck to the vault.
Yes.
Okay, and all of this we can put in a single piece of DNA such that when it gets transcribed, the vault protein gets transcribed to make the vault, and also the poly-A binding protein gets transcribed. You see?
That is so...
It makes the whole package.
We've created a vertically integrated stack at this molecular level